As we've discussed previously, there are lots of different levels at which you can look at ecological interactions. You can look at the organismal level, the population level, community level, ecosystem level, etc. But this chapter, we're going to focus on the population level. So we're going to look at how populations interact with their environment and how their environment can influence the size of a population, the density, the distribution of individuals, the age structure. Do you have more young or more old individuals? And all sorts of other things. Let's start by revisiting our definition of a population, because it's always been a little bit fuzzy. A population is just a group of individuals of a species, of the same species, living in the same general area. And we've talked about them in terms of population genetics, essentially as a gene pool, right? A group of individuals who can swap DNA, who might mate with one another and produce offspring. But again, this is a little bit of a gray area, but we describe them essentially by their size, their boundaries, and we can characterize populations by their density, essentially how many individuals are there per unit area, or volume if you're talking about water or air or soil, I guess, or dispersion, right? So how are individuals spaced? Are they clumped? Are they uniformly spaced? And as scientists, population ecologists have to find ways to measure things like population size and population density. And that's easier in stationary organisms like plants. So first of all, you could just count up every plant in a forest, and that is the total number of individuals in that population. Now that's difficult because forests are huge. Populations can cover large areas. So more often what you'll do is you'll take samples. So you'll go to different random parts of the forest, if it's a forest-dwelling plant like an oak tree or something, and you'll say, all right, in this plot, how many oak trees do we have? Okay, now we do this 100 times. We look at 100 different plots and say, all right, how many oak trees are in each of these plots? And then we get an average, and we multiply it by the total amount of land, and we estimate, oh, well, if there's an average of 3 oak trees per 100 meter squared plot that I measure, and then we just multiply that out by the number of meters squared in the total population. So that's pretty straightforward for stationary organisms like plants. For animals, it can be trickier for organisms that move, because you can't just count a plot and then move and count another plot, because that same animal might move and be counted again in another plot, right? Or they may run out of your plot while you're trying to count it. So for animals, population ecologists will do something called a mark recapture method. What this essentially means is you catch a bunch of animals, right? Let's say 100, right? And you mark them all. Every single one of them somehow. Could be a little ear tag, or could be a collar, or a little bit of paint, or for different organisms it's different. And then you release them, and you give them time to mix with the rest of the population. And then you go out later, once things are sufficiently mixed, and you capture another 100 individuals. And let's say that 20 of them are marked. That means that you probably marked about 1/5 of the population with your initial 100. So that means that the population is probably about 500 individuals, right? If you mark 100 individuals, release them, and then go back and capture 100 more individuals, and 5 of them are marked, that means you probably initially marked about 1/20th of the population. And so that means it's probably 20 times 100, right? Or 2,000 individuals in the population. Sorry, had to do math in the noggin real quick. Speaking of math, you can use this method and this simple equation here to estimate your total population size, or capital N. So capital N, your total population size, equals, again, we're estimating, but S, which is the number of individuals you initially caught and marked, times N, which is the number of individuals that you capture the next time in your sample, divided by X, or the number of those N individuals that happen to have a marking. So going back to our last math example, if we capture 100 individuals and mark them, that's S. Capture 100 individuals the second time, that's N. So 100 times 100 would be 10,000. And let's say 5 of those individuals in N were marked. So 10,000 divided by 5, that equals 2,000, right? So 2,000 individuals, that's our estimate of the population. For animals that move a lot, like dolphins, for instance, this is the best way to get an estimate of the sample size and the density, right, which is the number of individuals that you've estimated within the area that you are sampling. Population size and density are a function of things that can add or subtract individuals from a population that lives in a certain area. So what are those things? Well, essentially births and deaths, right? Births can add individuals, deaths can remove individuals from a population, and also immigration and emigration. And this is individuals coming in from other nearby populations or leaving to go to other nearby populations. So you can add to a population through births and immigration, and deaths and emigration can remove individuals from the population, making it smaller and less dense. The spacing of individuals within a population can depend on both social factors and environmental factors. For instance, if organisms show a clumped distribution, meaning instead of being sort of uniformly distributed in a population, you find a whole bunch of organisms right here or right there and very few in between. That can be caused either because of resource availability. There might be a bunch of food in that particular spot or in this particular spot. And so organisms clump around food or around shelter or protection, something like that. Or it could be because organisms are social. Social animals tend to show more of a clumped distribution. These are your three main types of dispersal patterns that you'll see in animals. We already talked about clumped, right, where you have a lot of individuals in a specific area and very few or no individuals in between these clumps. And that usually occurs because organisms are clumped around areas where there are resources or because they're social and they rely on one another and move in packs, like wildebeests or birds. Uniform distribution, where animals are very specifically separated from one another at very regular distances. This is usually found when organisms are territorial. So you can't get any closer than that because they'll fight you off. They're fighting for the resources in a territory. They also are not attracted to other individuals, right? So you have regular spacing when you have territoriality. Random distribution is what you find when there's no particular attraction to other individuals and also no aversion or territoriality. You just sort of toss the marbles on the table and they land where they land. You toss out your dandelion seeds in a field and they land and grow wherever they grow. Population ecologists will often study demography, or the vital statistics of a population and how they change over time. And in terms of vital statistics, there are none more vital than birth rates and death rates. One way to organize this type of data is with a life table. So you start with an age-specific summary of the survival patterns of a cohort. A cohort meaning a group of individuals of the same age. So everybody born in 2010 is in a particular cohort. And so we'll look in a moment at a life table for Belding's ground squirrels, and it will tell you a few things about the population. Here we have a life table for Belding's ground squirrels. It separates females and males. And so the first year, at age 0 to 1, there are 337 females alive at the start, 349 males, pretty close. And 207 females die and 227 males die. So a few more males than females. Death rate slightly higher, not a ton higher. And then we move on to the next year. So this is a weird number because it includes extra individuals that were caught. It really should be the number alive at the start minus the number dead. Should give you the next number. But anyway, so we start with 252 females, 248 males. And in this case, we have a death rate of 0.5 and 0.56 for males. And then 127 alive, et cetera, and you can keep looking at this. And you may notice that the death rate just keeps being a little higher in males, especially right around here, right as you get into the third or fourth year. Right, the death rate gets very high for males, to the point where after this fifth year, there are no more males alive. Some females are still alive. And you can plot this on a graph that we call a survivorship curve. and it can visualize how this information might be different. So here you can see the survivorship curve, so age over the number of survivors. This is a log scale, but you can see that females and males start off on a pretty similar curve, but then males tend to curve sharply downward, and females continue going at a fairly linear survivorship curve. There are three main types of survivorship curves: 1, 2, and 3. And yes, a lot of organisms fall in between here somewhere, but in terms of just understanding patterns in survivorship, this is a pretty useful way to look at it. So type 1 includes humans, and we tend to have relatively low deaths for most of our lives, but then as humans get older, we tend to have much higher mortality, right? People that are in their 70s, 80s, 90s, right, are much more likely to die in any given year than people who are 10, 20, 30, 40, 50, 60, etc. Type 2 survivorship curve means you are essentially no more or less likely to die at any age, right? You're just as likely to die at 1 or 2 years as you are at 5 or 6 years. And this is sort of normalizing these lifespans to make them equivalent. Obviously, humans live longer than squirrels, but any year that they're alive, they're just as likely to die as any other year. That's type 2, so it's very linear. Type 3, you have a huge die-off early on, but if you survive past youth, you have a very high likelihood of surviving a long time. So this x-axis is percent of max lifespan. So you find this often in like oysters. So think about giant oysters. These are born very, very tiny, and they float through the water, and they're very likely to get eaten or just die early on. But if they live, my goodness, they can live a long time. Also, sea turtles, right? Sea turtles, they lay their eggs on the beach, and then they all hatch, and they try to scurry off to the shallows, and they very often get picked off by birds or crabs or whatever when they're very young. But if they make it past those very early times, they get that very strong shell, they get very large, then they'll live for often over 100 years, right? That's a type 3 survivorship curve. So aside from death rates, birth rates are also very important in terms of population ecology. And so a reproductive table or fertility schedule is an age-specific summary of reproductive rates. It's much like a life table. It's also very useful, but it tends to focus on females in organisms with sexual reproduction because they're the ones that have the babies. Here's a look at Belding's ground squirrels. You may notice that this first year, this cohort of females have no babies. That means they're not sexually mature yet, right? But their second year, they are sexually mature. They're able to have babies, and they tend to have a relatively consistent number of babies, peaking at their 4 to 5 year, and then slowly going down from there. Other organisms may take years to become sexually mature, like humans or elephants or whales. That matters when you're looking at their population dynamics, right? An individual who's 2 years old can't start to reproduce and bring population numbers back. That said, Belding's ground squirrels continue having maybe not quite as many, but a good number of offspring through the end of their life, whereas other organisms, again, like humans and others, have an end time, right? Menopause is something that occurs in humans, and after mid-40s to 50s, women can't have babies anymore. And so knowing that helps you understand population growth dynamics of a particular species.